Hi, and welcome to our fourth week of the 3P seminar series. This series is all possible thanks to a collaboration between the Cure Parkinson's Trust, World Parkinson's Coalition, and the Van Andel Institute. My name is Michaela Johnson. I'm a postdoc in Patrick Brunden's lab at the Van Andel Institute, and I'll be chairing today's session. Our first speaker is Nora, a research fellow joining us from Oxford University, and today she'll be discussing how CLR01 protects dopaminergic neurons in vitro and in vivo in mice and human models. If you're new to Zoom webinars, if you look at the bottom of the screen, you'll see a Q&A feature. Feel free to add any questions you have for Nora throughout her presentation here, and when we get to the Q&A portion, we'll address these. Okay, over to you, Nora.
Thank you, Michaela. I'll start sharing. Right. So thank you for giving me the opportunity to share our data with you today. I'll be talking about the molecular tweezer CLR01 and how it protects dopaminergic neurons in vitro and in vivo in different models. So as an introduction, I'm just going to talk briefly about things that you may not know. So CLR01, depicted here in black to the left, is a molecular tweezer, which is just that, a molecule that looks like a tweezer. And its function normally works through stabilizing lysines, as you can see here depicted in blue. Now, if you think about lysines in blue and stabilizing them, you might worry that if you stabilize all lysines, this would be a very toxic molecule. But actually, it's not, because it's not a high affinity molecule. It doesn't covalently bind. It's a kiss and run type molecule with very low affinity. And so what it will do will be to actually just briefly touch on those lysines. And it won't interfere with your regular cellular processes, and that's what makes it very safe. It will only deal with overrepresented processes, such as amyloidogenic processes. And so we thought that investigating its effects in the context of Parkinson's disease would be really interesting. I also want to talk a little bit about synuclein oligomers, because I'm sure that you're all familiar with Parkinson's disease and alpha-synuclein, but you might know a little bit less about oligomers because they're a little bit understudied when we compare them to monomers or fibrils and other lesions. So if we think about oligomers and a neuronal cell in the brain, these interfere with many of the pathways that are important to a neuron. We think about autophagy or lysosomal function. They can block this, causing accumulation of oligomers and other detrimental species. They can cause ER stress and dysfunction, proteasomal dysfunction, and therefore impair all the waste management of the cell. If we look at the mitochondria, they can cause defects in fragmentation, causing influx of calcium and complex function, and therefore inhibiting the ability of the cell to produce ATP and power for what it needs the power for. If we think about the synapse, it can cause impaired vesicle trafficking and abnormal synapse recycling. If we think about the membrane, it can punch holes into it, and then which leads to ions getting in and out and going where they should not belong. And finally, outside of the cell, it can also cause inflammatory response by activating microglia and other cells not depicted here, other immune cells and even astrocytes. And so why they've been under research is that it was very difficult to detect them. But in 2015, Rosie Roberts in the lab developed the alpha-synuclein proximity ligation assay, or ASPLA for short, which actually helped us visualize these oligomers and these tiny puncta here, not these big ones, those are nuclei, but the tiny puncta that you can hopefully see there. And what she could show was that there was previously unrecognized pathology in the PD postmortem brain, in regions of the brain that you could normally not see a lot of Lewy pathology in, such as the cingulate gyrus. And we've gone on to validate this further in the striatum and the hippocampus. Finally, I'd like to introduce the two models that I'm going to present results on. iPSC-derived dopaminergic cultures, shown here to the right-hand side. We use iPSC cells, or induced pluripotent stem cells. We derive them, differentiating them into the dopaminergic lineage through the CRIX protocol of the Studer lab. And we get these beautiful neurons that are Th positive, and we can use to model the human brain. And on the other side, we can see our synuclein overexpressing animals, or synuclein OVX for family and friends, which were developed by a very talented student, Steffi Janesek, in the lab. She basically took a synuclein knockout mouse, so a mouse that has no murine synuclein, and she packed the whole human synuclein locus into these animals and overexpressed it. What she could observe was that at 3 months of age, the animals were pretty normal. There was no significant differences in rotarod behavior or in the number of Th cells in their nigrus. But when she aged them, she could see that synuclein OVX animals performed significantly worse on the rotarod. She could also see that neurons were lost in synuclein OVX animals, and that was not the case in their litter mates. So she detected an age-dependent dopaminergic degeneration, which mimics that in Parkinson's disease. But she also detected some effects that were there from the younger age of animals. So at 3 months and through the whole lifespan of the animal, she could see a dopaminergic release defect in the striatum as measured by FCV. And so she has a model, or she made a model, where we can have aggregation and age-dependent defects that I'll talk about a bit later, but also defects that are probably more due to the monomer and the overexpression of the protein itself. So I'm just going to plunge into results. The first thing that we did was to start in vitro, looking at our iPSC cultures and insulting them with Lewy body derived extracts, for which we teamed up with Erwan Bazard in France, in Bordeaux. And they're specialists in producing these Lewy body extracts from Parkinsonian brain. And they purify them out, and they detect the Lewy body fraction. They ship it over to us, and we insulted our cells with it in the presence or absence of CLR01. So the first thing that we could see was just macroscopically, you can see a colony of dopaminergic cells here with their processes. And once you insult the cells with Lewy bodies, it becomes very turbid. With the texture. These suspensions are more suspension than a solution. The Lewy body ones, they're quite a cloudy one. And already macroscopically, CLR01 was able to remove that cloudiness. If we look at alpha-synuclein oligomers, and we quantify them here in panel B to the right-hand side, we can see that there's a very modest increase when we treat these cells with Lewy body material, and that when we treat them with the tweezer, we can reduce this. We were not surprised by the modest effects of the Lewy body material on alpha-synuclein PLA signal, because if you look at postmortem human brains, PLA classically decorates the Lewy bodies. On the outside, it will show a ring of dots around the Lewy body lesion, but it won't stay the Lewy body lesion, indicating that the oligomers that we're detecting by ASPLA are probably outside crowning the Lewy body and perhaps contributing to its formation, but definitely not in the inside core. And so it's not a surprise that they're not terribly enriched. But what is far more interesting is if you look at these panels, when we did process analysis, you can hopefully see on your screen that there's a nice process there and there in control cells. Same here or there for CLR treated cells. But if we look at Lewy body treated cells, there's only vesicles left or dots left where the processes would have been. And that's because axons have started blebbing, they've started degenerating as an early sign of unhappiness in the culture. And we can automatically actually quantify this as the process length, which is reduced when we treat cells with Lewy body extracts, and we can rescue this with our molecular tweezer. And the same, if we count these vesicles and the roundness, we can actually give them a score for blebbing, and we see that there's an increased blebbing in the Lewy body treated cells and a reduction of it in the tweezer treated cells. So because of these results, we decided to go and explore the effects of the tweezer a little bit further. And what we could see, what I'm going to describe first is some phenotypes that we've worked on ever since Steffi published her original paper in 2013. That I'll also be looking into with the treatment of the tweezer. So if we think about astrocytes, we look at wild-type animals, they don't seem to actually differ a lot with age, but if we look at the OVX, we can see that at 12 months of age, they start having a little bit of an increased staining for GFAP in the nigra. If we look at microglia, we cannot see differences in cell number, but we can see differences in morphology. We can see that in wild-type animals, they are pretty much fine. But in OVX, as soon as we hit 6 months, we can see a shift in the population, and they go a bit more amoeboid and a bit less ramified. So quite early effects, we think probably due not to aggregation, but perhaps more to the overexpression of the monomer and then reacting to the monomer. And finally, we quantify through time PLA and wild types. There's a little bit of an increase, but the magnitude is tiny. And when we hit 9 and 12 months, that's when we start seeing quite nice levels of aggregation that will significantly or reproducibly be detectable in our cohorts. So we think again that we have this phenotypes that probably come in earlier and are more to do with the monomer, and then phenotypes that are more to do with the aggregate that come in a little bit later. And so we decided to treat these transgenics at 6 months, 12 months, and 18 months, thinking that 6 months would be quite happy and healthy, non-symptomatic animals. At 12 months, hopefully, we'd see animals that are perhaps on their way, perhaps on the prodrome, navigating those early stages of pathology, and then at 18 months, animals that are already undergoing neurodegeneration. And so at 6 months, we cannot really elicit any effects with CLR01. If we look at pathology to begin with, we can't revert the amount of pathology that is there. We also can't always significantly detect it at 6 months, to be honest, because aggregation is quite variable and low there. If we look at dopaminergic release, here in the dorsal and ventral striatum, we can see that there's no effects of the drug in our animals. And the same for microglia. So we think for monomeric driven effects, which are probably the ones that we have at 6 months, we can't really do much with the tweezer. However, when we go animals at 12 months, what we can see is that there's a very minute, but albeit significant effects, 10% defects in rotarod and catwalk analysis in the OVX when we compare them to wild-type animals. And when we treat the animals with the tweezer, we can restore these effects. When we look at the midbrains of the animals, we can see that, again, there's an accumulation of oligomeric pathology in dopaminergic neurons, which we can revert by treating the animals with CLR01 for 2 months. And if we look at the glia in the nigra, we can see that GFAP is increased by the astrocytes and then again reduced by CLR01. Funnily enough, we were expecting the microglia to follow suit and then perhaps go into a more ramified and resting state. But indeed, they went a little bit more amoeboid. And our current hypothesis for these results is that because CLR01 will inevitably release dissociated species into the environment, that the microglia are probably reacting to these and actually going into a phagocytic mode where they're munching down on the species that we're releasing. We're currently investigating this further and looking at whether it's actually true in our animals. Finally, we treated 18-month-old animals where degeneration has already been present and it has set on. And I haven't brought the non-significant graphs with me here today, but we can't really affect motor defects at 18 months. We cannot restore them with CLR01, which wasn't a surprise because we're not looking to bring neurons from the dead with the molecular tweezer. We're looking to actually treat pathology, early pathology, as soon as it hits. So when we look at pathology, we can see a downregulation of oligomers in the substantia nigra neurons, astrocytes, and microglia, and an upregulation of markers that have been associated with positive effects in Parkinson's, like PGC1 alpha, which is a master regulator of mitochondrial biogenesis, and LAMP2A, which is a mediator of chaperone-mediated autophagy. So we think, taking all these cohorts together, that the important message is that one needs an optimal window of opportunity, and one needs to know, in my case, I need to know my model, but we also need to be able to translate this to humans. We need to know what the pathology looks like and when the prodrome starts, and in order to be able to accurately treat people. Now, the final two experiments that I'm going to show are injection paradigms, because we wanted to address whether alpha-synuclein higher molecular weight species were also affected by CLR01 treatment, or whether CLR01 was only able to address oligomers in vivo, because we know in vitro that it works on fibrils, etc. So here we have the Kelvin-Loken-Virginia Lee PFF injection model, where I injected PFFs into the striatum, dorsal striatum of the animals, and therefore have more lesions in the SNC than in the VTA. And we can see that there is more phosphorylated lesions in the SNC, and that CLR01 is able to actually downregulate these effects. And again, in collaboration with France, Emily Fadiani did this work on the LB injection model. They have shown, again, in mouse and non-human primates, that LB material can cause degeneration of dopaminergic neurons. So she injected these into mice, and what she could see was that after the injection, she could rescue the effects by dosing the animals for a month with CLR01. She could also restore innervation to the striatum and reduce PK-resistant alpha-synuclein lesions and lesions stained with the SYNF1 antibody. So to conclude, I'd like to leave you with the thoughts that CLR01 is able to revert the toxic effects caused by Lewy body extracts on iPSC-derived dopaminergic cultures, that it's able to correct age-related neuropathology and motor defects in a synuclein overexpressing model, but the timing is critical, and that we really, really need to define this in the human, because otherwise, even if CLR01 was ready for the clinic tomorrow for safety, etc., we couldn't really know when was the right time to treat these patients. And finally, it is able also to rescue neuropathologic effects of 2 different intracranial injection models. I'd like to thank the people involved in this study, people from our lab, Ejim, Natalie, Steve, Melina, and Richard, and everyone else, Emily, Benjamin, and Juan from Bordeaux, Paula, Fabian, Carlos from Spain, Sarah, Bradley, and Steph from over the road in Oxford, and finally, Thomas, Frank, and Gal for making the tweezer, and you all for your attention. Thank you very much.
Thank you, Nora. Fantastic presentation. We'll kickstart the Q&A session now. First off, we have a question from Vele. Could you give us some details on the mode, dose, and time of administration of the CLR01 in vivo experiments?
Sure. So for all cohorts, really, except for the 18-month-old transgenics and the last injection model, the Lewy body injection model, we have used subcutaneous injections, 16 subcutaneous injections over 2 months at 40 kgs per day, 40 mg per kg per day, if that makes sense. For the other remaining cohorts, we treated both the 18-month-old animals and the Lewy body injected animals in the Niagara with osmotic mini pumps, which essentially at the same dose and everything. It's just that they have continuous administration rather than giving them bolus of 16 different injections Monday and Thursday.
And I guess a follow-up question to that, do you know how much of that compound, when it's given systemically, reaches the brain?
Yeah, we're currently actually building a model in silico to model up to the cell how much is coming in. Whatever is available in the blood, you'll get 2% of that roughly in the brain. So that's really interesting. But what's more interesting is that cells are actually really efficient at taking it up. We don't know why, perhaps because it binds so loosely, but it binds to proteins. We know that it's via endosome-lysosomal pathway, so maybe it just kind of just gets there, binds to a protein, and then it gets trapped into the endosome-lysosomal system. So yeah, we're building a model to see, even from the 2% of the blood, how much would be the factual concentration inside the cell.
Excellent. So we'll find out more about that at a future date. Question here from Amanda. Do you know what the actual mechanism is for the clearance of pathology by this CLR01?
I guess you mean how it dissociates the aggregates. The way that it dissociates the aggregates is because we tend to think about aggregates as in things that are very static. We think about fibrils, and they're a big fibril, and they never dissociate or associate. But you have to think of them a bit more like a microtubule, which is all the time constantly associating and dissociating. They're plastic, they're dynamic. Even if you flash freeze them, you'll get different species. No one has ever produced a fibril that has no monomer trace with it, because it's constantly associating and dissociating. The way that CLR acts is by sneakily perhaps just joining into the lysines when it gets an opportunity. And you do have to dose it quite highly because, or have a high local concentration of the tweezer, because otherwise you don't have enough tweezer per lysine, if that makes sense. The other interesting thing about synuclein is that the region that is central for aggregation for it, the NAC domain, it's completely ribbed with lysines.
Okay, so I think that also answered another one of our attendees' questions, Maddie, who was asking about whether it was directly interfering with the formation or the stability of those alpha-synuclein oligomers.
Mm-hmm.
A question more about the behavioral side of it from Poonam. Nice talk. Could you please explain the catwalk behavioral test?
The catwalk that we use is the Induced one. So I know that people kind of have also done it a bit more manually by having the mouse put on ink and then letting it run on a paper trail and actually measuring. But we have a nice automated system that has a red light as background and then a green slide, if you will, and a camera underneath. So what happens is the animal, as it walks, we'll see its green paws on the red background, and we can automatically detect all of the parameters in gait. So we can measure basically everything from the size of the paw to how long the step was, how long it took him to get to the next step, etc.
Excellent. A question about the induced pluripotent stem cell work here from Naveen. For those experiments where you use the derived dopaminergic neurons, what % of neurons were TH positive, and what protocol was used to achieve this?
We use the CRIX protocol from the Stuart lab. I think there's slight variations, but if you go on any of our latest Waite-Martin's paper, they'll be on the detail or whatever we change is on there. So, and they're very minimal. We do get variability in the cultures. Sometimes we get cultures that are very enriched for Th positive neurons. I've had lab mates have up to 70% enrichment for dopaminergic cultures. I'd be a bit more humble and err on the side of caution, maybe say 40 to 50% of dopaminergic cultures. And what I do like to do within my experiments is to QC the cultures at 25 days. If they're within parameter ranges that are comparable, then they are taken forward for experiments. If not, I just don't, because no point in comparing different things, I don't think.
Yeah, definitely. Okay, another question here from an attendee. Do you have information on CLR01's effects on neuropathology and other cell types other than the nigral dopaminergic neurons, or do you plan on looking at that in the future?
We did do it for the 18-month-old animals. I have to confess that I haven't done it for the, well, not for the 6-month-old, because there's no reduction in pathology. So it's I mean, you could show the non-reduction in many different cell types, but it would be a bit, perhaps not time well spent. I haven't done it for the 12-month-old animals, but it would be something that we could do. What I usually see is that if it's down in Th cells, then you'll see it down in the astros and down in the microglia. So I kind of put those experiments a little bit to the side because they do correlate quite nicely.
Thank you. Question from Sarah: which alpha-synuclein antibody do you use for the ASPLA? And can you differentiate between oligomers and their fibrillar aggregates using that assay?
Yes, so we use, we've developed 2 different assays. The paper by Rossi was using the antibody 211 from AppCam, and it was specific to oligomers. When we do assess monomer, oligomer, and fibril in recombinant species, we do see a little bit of detection in the fibril, but we're always left wondering whether that's because there'll be a little bit of oligomers in fibril preps. It's inevitable, and vice versa. And the same way that there's a monomer in all preps, et cetera, et cetera. And so the same is true for the new assay that we use that is a bit more sensitive and specific, which uses the, again, AppCam alpha-synuclein 4D6 antibody. I believe the reference is AB1903. And again, we see the same thing. We see an enrichment, a high detection power for oligomers, and a bit of detection for fibrils. But again, we're left wondering whether it is because of the mixed population within the preps and that inability of us to get pure 100% fibrils ever.
Thanks. Another question here from Mehtat: how specific is the CLR01 to alpha-synuclein pathology? Would it work with other aggregation proteins like tau or amyloid beta?
Yes, that's a great question. It will definitely work on other proteins. There's previous work on the triple transgenics for Alzheimer's disease on the mice, showing that it can clear up tangles and plaque, I believe, and then reduce gliosis as well. So anything with a lysine, it'll see. The only thing is that unless, well, if you overdose an animal, then of course it'll end up seeing every process in the cell, and you potentially can saturate it, but you'll need insane doses to do so. But if you look at pharmacological doses, then it'll only see things that are really overrepresented in the cell, and still things that need to really be clumping around, for lack of a better word.
Yeah. So I guess one final question is, where do you see this research going? What's the next steps for this potential treatment option?
That's a great question. There's other similar compounds being looked at for in the clinic, but CLR01 has not really gotten there yet. I think it'll probably need a little bit more work on the compound to make it to the clinic. I think we would need to address availability, because 2% is still quite little. So that's why we're also working on the in silico model to see how much will be present within brain cells, because that's quite important. So I think that there's a little bit more preclinical work to be done, but surely we do kind of need to start thinking about what aspects will drive it to the clinic. And definitely we need to work more on the timeline in the human to determine when the equivalent to the 12-month-old animals is, because otherwise we can potentially liberate many or free up many harmful species within Lewy bodies, which we wouldn't be keen on doing, or neurons might have already been too damaged to actually be rescued.
Excellent. So we'll keep our eyes peeled for looking at this in the future. Thank you for an excellent presentation, Nora. There's a few questions left, but if you want to continue the discussion using the 3P discussion Facebook group, we'll post these questions there.
Okay.
Thank you again.
Thank you.
Our second speaker for today's session is Anke Van der Perren, a postdoc at KU Leuven. She is presenting on the in vivo characterization of distinct patient-derived alpha-synuclein strains. She's presenting unpublished data, so can everyone please refrain from taking any screenshots or photos during her presentation. And on that, again, please use the Q&A feature during this session for any questions you have for Anke. Over to you.
Okay, good afternoon, good evening, everybody. So I will today talk about the in vivo isolation and characterization of distinct patient-derived alpha-synuclein strains. And let's start with that. Okay, so we know that the protein alpha-synuclein in its aggregated form is a key player in Parkinson's disease, but is also very important in other neurodegenerative disorders like dementia with Lewy bodies and multiple system atrophy, called together synucleinopathies. These disorders display distinct clinical symptoms, like Parkinson's disease is characterized by Parkinsonian motor features, including postural instability, rigidity, tremor, and bradykinesia. DLB has a similar clinical manifestation, but cognitive symptoms typically occur 1 year before the onset of the motor symptoms. And in MSA, we have mainly autonomic dysfunction combined with Parkinsonian features. These different disorders have also different histopathological hallmarks. In Parkinson's disease and in DLB, alpha-synuclein mainly forms large neuronal inclusions called Lewy bodies and Lewy neurites, whereas in multiple system atrophy, alpha-synuclein aggregates are mostly found in oligodendroglia as glial cytoplasmatic inclusions, or GCIs. So different diseases have one key player, namely alpha-synuclein. But so this raises the question, how can one protein induce different diseases? Is this due to the environment of the protein, or might there exist different structural synuclein conformation or strains? In 2013, our close collaborator, Professor Ronald Melky, was able to produce different recombinant alpha-synuclein strain in vitro using different assembly conditions. Under different cell conditions, he was able to assemble, for example, alpha-synuclein fibrils, and under low cell condition, they produced alpha-synuclein ribbons. They have shown that these different fibrillar forms have a different toxicity, seeding, and propagation properties in vitro. In 2015, we extended this data in vivo, and we have shown that different recombinant alpha-synuclein strains also induce a distinct phenotype in vivo. Here is a brief summary of what we have shown. So we have shown that alpha-synuclein fibrils induce a higher level of neurotoxicity, and also this was paralleled by motor impairment, whereas the alpha-synuclein ribbons induce more Lewy body pathology, and it also induced in a small amount the presence of JCI-like inclusions, which is more a characteristic of MSA. But so from this study, we could conclude that different alpha-synuclein strains cause distant synucleinopathies in vivo. But a big question brings us that how are these different strains, are they really present in the human brain of individuals suffering from PD, DLB, or MSA? And so this is something we wanted to investigate in the study that I will present today. Together with the Professor Ronald Melky from the CNRS in France, we have set up a large study where we isolated Lewy body-rich regions from PD, DLB, and MSA patients, as well as age-matched control. From the PD and the DLB patients, we isolated the cingulate cortex, and from the MSA, we isolated the cerebellum. Each group contained 4 to 6 patients. Next, we used an in vitro method called PMCA, or protein misfolding cyclic amplification, to amplify the different human-derived alpha-synuclein strains. These different strains we then characterized in vitro and in cell culture, and then in the end, together with the total brain homogenate, we injected these PMCA amplified strains in vivo in the midbrain of rats, or naive rats, or rats overexpressing alpha-synuclein. First, as I said, we made brain homogenates and assessed the amount of alpha-synuclein, the amount of total alpha-synuclein, and the amount of phosphorylated alpha-synuclein. You can see here from the quantification and also from our filter trapping assay that there is a big variation between the different patients. Here you can see it for PD, for MSA, and for DLB. And here is the quantification of the total alpha-synuclein and the phosphorylated alpha-synuclein. But what was remarkably was that the amount of phosphorylated alpha-synuclein was almost not present or very low in the control cases. Next, we used these homogenates as seeds to amplify the specific human strains in vitro. We did it with an PMCA assay, and so the human material is a seed. We add the monomeric alpha-synuclein, and like this, we amplify the human-derived material. We use 3 amplification cycles, which you can see here. And then the amplified material was further biochemically characterized. First, the alpha-synuclein fibrillar assemblies were imaged by transmission electron microscopy, and we observed disease-specific differences in the shape of the fibrils. If you look in detail, you can see that the Parkinson and the MSA fibrils, that they resemble a relatively flat and twisted appearance, which was resembling the ribbons, recombinant ribbon conformation, and that the DLB-derived fibrils had a more cylindrical without twist conformation, and this looked more like the recombinant fibrils conformation. So clearly there was a difference in structure between the PD, MSA group on the one side and the DLB-derived fibrils on the other. Next, we assessed their limited proteolytic patterns using exposure to proteinase K. So we ran an SDS page after proteinase K treatment, and so we saw that again the PD and the MSA derived strains displayed a different pattern. As you can see here, there are 2 bands present, whereas the DLB derived alpha-synuclein fibrils presented a different pattern. You can see here that there are multiple bands present. Again, the PD and the MSA displayed the profile we observed with the recombinant ribbons, whereas the DLB displayed the profile that we observed with the recombinant fibrils. Last, we also used the conformational antibody PHILA-A4, and we saw that all the PMCA amplified patient-derived alpha-synuclein fibrils were recognized by the antibody, but to a different extent. You can see here that the 4 DLB cases were much better detected than the MSA and the PD and the MSA patients. And here you can also see the quantification. So from this in vitro characterization, we can conclude that alpha-synuclein fibrils derived from one synucleinopathy have a comparable fingerprint and morphology. So all our patients within one disease group had a similar fingerprint. But when we compare the different groups, we can see that PD and MSA fibrils have a similar shape and limited proteolysis pattern, and that it was different from the DLB-derived fibrils. Next, we assess the seeding capacity of the distinct PMCA amplified alpha-synuclein strains in a human H4 neuroglial cell line expressing alpha-synuclein YFP. We use recombinant fibrils as a positive control, and so upon exposure of the H4 cells to all the different strains, after 24 hours, we saw a clear aggregation, as you can see here in the dots, and a more detailed picture over here. And we quantify this as the number of YFP positive puncta per H4 cell nucleus, as you can see here. Also, we observed that the aggregated alpha-synuclein colocalized with the phosphorylated alpha-synuclein. And we saw that the control conditions didn't induce any aggregation. Also, the homogenates didn't induce any aggregation, but when we compared the different disease groups, we clearly saw that the DLB group had a higher number of YFP positive puncta. And if we look to the individual patients, you really see that 2 out of the 3 DLB patients were significantly positive compared to all the MSA and PD patients. Next, we also assessed the morphology of the aggregates. Because we saw some differences. We saw that our recombinant fibrils had a much reduced, smaller width to length ratio, as well as a roundness compared to all our experimental condition. But then when we looked into detail, we could also observe that the DLB conditions had also a smaller width to length ratio and a smaller roundness compared to all MSA and PD cases. So the cellular assay clearly displayed that the DLB had more aggregates, but they were clearly smaller. Then we also exposed primary neurons to our different strains, and we could see that after 7 days, we clearly had the formation of phosphorylated alpha-synuclein deposits. And we saw that there was a distinct morphological pattern. In the PD cases and the MSA cases, we observed 2 different types of phosphorylated alpha-synuclein neurites. On the one hand, we had these thicker linear neurites and this more dotted phosphorylation pattern. So both in PD as in MSA. Whereas in the DLB group, we had a more diffuse alpha-synuclein deposits within the soma surrounding the nucleus, as you can see here. So from our cellular assay, we conclude that the PMCA amplified alpha-synuclein strains see the aggregation of alpha-synuclein to different extent and induce different phenotypic changes. To determine whether the different characteristics of the alpha-synuclein strains would also be present in vivo, we inoculated both the brain homogenates and the PMCA amplified assemblies, so the pure material, into the rat substantia nigra of naive and alpha-synuclein overexpressing rats. First, I will present you the data of the rats injected with the alpha-synuclein, well, also with the total brain homogenate. And so first we followed up motor deficits in the animals over time, up to 5 months by the cylinder test. In naive rats, as you can see here, there is no difference in motor deficits. When combining the brain homogenates with alpha-synuclein overexpression, we observed that there was a clear increase in motor deficits and that this was most severe in the MSA group. Next, we assessed the level of dopaminergic neurotoxicity in these animals injected with an AAV vector combined with the homogenate. And we saw that the most severe nigral neuronal cell loss in the animals was present in the animals injected with the PD and the MSA homogenates. Also, when we assessed the percentage of striatal lesion, we saw that this was most significant in the MSA treated animal. If I then go over the data of the rats that were injected with the alpha-synuclein strains, again, we observed that there was no effect or no motor deficits present when the PMCA material was injected as such. When we combined it with the viral vector overexpressing alpha-synuclein, we saw that all the conditions, so the AAV, the control, the PD and the MSA had similar motor deficits, but very unexpectedly, we actually could not observe any motor deficits in our DLB injected animals. To make sure that no mistake was happened, we actually repeated the whole experiment, so the 5-month time experiment, but we obtained the same results. To check this further in detail, we analyzed again the neurotoxicity in the substantia nigra and the striatum. And so again, in the animals injected with the viral vector and the PMCA strains, we could see which is in agreement with actually the homogenate data, so that both the PD and MSA induced the most severe nigral lesion. And when we look to the effect in the striatum, so the percentage of striatal lesion, actually we see that the DLB group had almost no striatal lesion, which is actually in line with the absence of the motor deficits 5 months post-injection. Next, we also assessed alpha-synuclein pathology. And so first we analyzed the number of phosphorylated alpha-synuclein positive nigral neurons. When we assessed the animals injected with the total brain homogenate, we saw that the number of nigral phosphorylated positive cells was significantly higher in the MSA group compared to the other groups. When we analyzed the PMCA animals, so the PMCA injected animals, we saw that actually the DLB group had the lowest number of phosphocin positive nigral neurons, which is again in line with the absence of the motor deficits and the absence of the striatal lesion. Further, we also examined the presence of alpha-synuclein pathology in other cell types, such as astrocytes or oligodendrocytes, but we've only found alpha-synuclein positive neuronal inclusions. So you can see here. So here are clearly the TH positive neurons, but no GST pi, so which is a marker for the oligodendrocytes, are positive for phosphosynuclein. And we also found alpha-synuclein positive neurons in the striatum, so in the projecting region, as these were positive for DARP32. Next, we also assessed the distribution pattern of pathological alpha-synuclein within the brain. If we then first look to the homogenates which were injected combined with the viral vector, we clearly see that our MSA conditions gives the most distribution of the pathology. So we have pathology present in the striatum and the connecting regions. If we then look to the PMCA injected material, we see that there is more pathology present. and that it's again the highest level in the MSA injected group, and very striking, again, the DLB group, it's almost, so alpha-synuclein pathology is almost absent in the other brain regions. The next thing we wanted to do was to investigate whether distinct alpha-synuclein strains may act as antigen, altering an immune tolerance. And therefore, we looked for the presence of different immune-related cells in the brain. In general, we could say that the immune response to PMCA-amplified alpha-synuclein strains was higher compared to the immune response to the total brain homogenates. When we then comparing the different diseases, it was quite clear that the MSA-derived material induced the strongest immune response, both for the homogenates as for the PMCA-amplified material. We saw a higher number of IBA1 positive cells in the MSA group. We saw also an increase in MSC2 expression. And then also we examined the infiltration of T cells, so CD4 and CD8, which was most highest in the MSA group, as well for the PMCA condition. So if I want to summarize the in vitro data that I just presented, I'm going to do this with this table. So I think we can say that for the total brain homogenates combined with the AAV viral vector overexpressing alpha-synuclein, the MSA group had the most significant motor deficits. We saw the highest level of neurodegeneration in the MSA, followed by the PD cases. We had the most severe striatal cell loss, a high level of alpha-synuclein pathology in the nigral neurons, and the most spreading of distribution of the alpha-synuclein pathology. And also the immune response was the highest in the MSA group. When we then compare this with our PMCAs, or our pure alpha-synuclein strains, it was very striking that we didn't observe any motor deficits in the DLB animals. Again, the MSA and the PD group induced the strongest nigral neuronal loss. Also, the alpha-synuclein spreading was most prominent in the MSA group, and also the immune response. Here we can conclude that similarities but also differences for the different phenotypic readouts were observed between the homogenates and the PMCA fraction. Of course, we have to mention that total brain homogenates are very heterogeneous in nature, so they contain, next to aggregated alpha-synuclein, a mixture of protein and probably inflammatory components. And so this is different than the PMCA assemblage, which represent actually the pure alpha-synuclein protein. So to conclude, I want to give you the highlights of our study. So we described a specific signature for patient-derived alpha-synuclein strains. And so we have shown that alpha-synuclein fibrils have distinct structural threads characteristic of each synucleinopathy. The PMCA-amplified alpha-synuclein strains see the aggregation of alpha-synuclein to different extent. Both the homogenates and the PMCA strains reflect the characteristics of the different synucleinopathies, and the MSA strains are the most aggressive, but also most similar to the PD-derived strains, and in contrast to the DLB assemblies. The differential immune response towards distinct alpha-synuclein strains shows that the synucleinopathy strain represents an unique antigen, and I think this is something we should check further in detail. And I think investigating different brain regions may also be relevant, since regional variation was described for different synucleinopathy. Last, and I wanted to say that all the data that I have presented here, I didn't give all the details from all the experiments, but our manuscript was just accepted for publication. So I think in 1 or 2 weeks, you can find our study online. And so last, I would like to thank the people from our group, because of course, these data, I haven't done all the work alone. I would especially thank Geraldine Gelders, who is a PhD student in our lab, and my professor, Professor Bakeland. Also our collaborators from the CNRS, Professor Ronald Melky, Luc Bousset, and Alexis Fenny, and also Professor Steve Gentleman from the Imperial College. And of course, all the people from our group and all our funding agencies. And of course, you for your attention. Thank you.
Thank you, Anke. We'll kick off the Q&A session now. So first question from Neil. Any chance the YFP construct interferes with or alters the aggregation dynamics of alpha-synuclein?
I mean, I think it's something we, of course, cannot exclude. That's correct. But yeah, this construct, well, it was expressed in the cells that were treated with all the different alpha-synuclein strains, and still we clearly observed differences. I think what is also interesting is that in our primary neurons, we didn't overexpress any alpha-synuclein, and there we also clearly observed differences between the different experimental conditions.
Yes. A question from Louis. Do you think the PMCA data is correlated with the phosphosynuclein deposits? DLB seems to form different phosphosynuclein deposits than PD and MSA.
No, indeed, it's true. I think it's quite interesting. Some things we cannot completely explain yet. I do think that, I mean, we have clearly amplified certain strains by using the PMCA method. Of course, the phosphorylated alpha-synuclein is a marker for pathology, but as you also can see, for example, in when we made the crude extracts from the brains, it was very variable between the different patients. So I'm not sure if these readouts will tell you everything.
A question from Pinam. For the DLB groups, did you have any behavioral tests for memory deficits that you might expect with this phenotype rather than the motor phenotype?
I think that's a very interesting question. I think for that, the model that we used is maybe not ideal to answer that question, because of course we used a model where we inject the different strains into the midbrain, so in the substantia nigra. But I think it would be very interesting to, for example, inject the strains in different locations, and then you could also observe and check different readouts as well.
So where do you think would be the most relevant brain regions to inject for MSA compared to DLB and PD?
Well, I think maybe for MSA, it would be very interesting to inject in the striatum or the cerebellum. And for example, I think for DLB, the neocortex would be also a very good location.
Interesting. A question from Amanda. Given that the fibrils from PD and MSA had the same shape and proteolytic pattern, but the response to the MSA injected mice is much more pronounced, what do you think is giving that different response in those MSA patient-derived fibrils?
Again, I think it's a very good question. We don't know. I mean, of course, the readouts that we use, so the TEM, the limited proteolysis, the conformational antibody, I mean, there is only as much as we can see and check a base for the structure at this moment. So I'm not sure if there are maybe subtle differences that we cannot see, because, yeah, there has to be or a difference, or it might be that based on a location, a strain can also be different.
Yeah, interesting. Well, thank you for a very interesting presentation. We have a couple of questions that will get posted into the 3P discussion group on Facebook. So please refer to that if you'd like to continue the discussion further. And I'll now pass it over to Lisa to talk about what we'll be presenting later this week.
Thank you to our speakers for that very interesting session. I learned a lot. And on Thursday, we're going to be joined by Maria Elena Sicardi, who will talk about the role of extracellular vesicles in a C9 ALS model. And then Sonia George, who's a co-worker to me and Mickey at the Van Andel Institute, will talk about if T cells influence alpha-synuclein aggregates in a mouse model of Parkinson's disease. And after that, we have more interesting sessions coming up. So keep an eye on the CPT webpage or on the Twitter account for 3P for more information. So thank you all for attending today, and I hope you have a nice rest of your day or good evening. Bye.
Bye. Thank you.